Welcome to this lecture on sequence similarity and homology. Imagine you are a teacher grading two essays. If both have the exact same unusual sentences, you would immediately suspect they share a common source. In computational biology, we do the exact same thing with DNA and protein sequences to understand their history and function. Building on that essay analogy, let us look at why sequence comparison is so vital. When we find a brand new sequence in a newly discovered organism, we desperately want to know what it does. Instead of starting from scratch in a lab, we compare it to a massive library of known sequences. If it matches a sequence that digests sugar, we can confidently guess our new sequence does too. This is also critical for drug discovery, allowing us to test treatments on model organisms that share similar genetic sequences with humans. Now that we understand the purpose of comparing sequences, we must strictly separate what we can physically observe from what we logically infer. First, we have sequence identity. This refers simply to the exact one-to-one matches between two aligned sequences. It is like finding the exact same letter in the exact same position across two different texts. Because we can count these matches directly, sequence identity is a strictly observable, measurable quantity that we always express as a percentage. Building directly on identity, we have sequence similarity. This is a broader term. It includes all of those exact matches we just discussed, but it also adds conservative substitutions. A conservative substitution is a change where one amino acid is replaced by another that has very similar chemical properties, like size or charge. Because these substitutions usually do not break the function of the protein, we count them. Just like identity, similarity is an observable, measurable quantity that we express as a percentage. While identity and similarity are percentages we can measure, this brings us to a completely different concept: homology. Homology is not an observation, it is an evolutionary conclusion. When we say two sequences are homologous, we are stating that they share a common evolutionary ancestor. Because of this, homology is strictly a binary condition. Two sequences either share an ancestor or they do not. Therefore, it is completely incorrect to say two sequences are 80% homologous. To quantify exactly how much two sequences resemble each other, we use a simple mathematical formula for percentage identity. Conceptually, this comes from aligning two sequences side by side, sometimes inserting artificial gaps to maximize matches, and then finding the ratio of exact matches to the total length. In this formula, I represents the percentage identity. N sub ID stands for the total number of exact identical matches. L represents the total length of the alignment, which includes matches, mismatches, and gaps. If the number of exact matches increases, the overall identity percentage naturally increases. Now that we have our formula, let us see this with a concrete example. Suppose we have two short protein sequences aligned in a lab. Sequence A is MVLTG, and sequence B is MILSG. We are asked to find the percent identity. First, we count the total length of the alignment, which gives us a denominator of 5. Next, we count the exact letter-for-letter matches. We have M, L, and G matching exactly, giving us a numerator of 3. Dividing 3 by 5 gives 0.60. Multiplying by 100 gives us a final sequence identity of 60%. Physically, this means 60% of the sequence has remained completely unchanged. Having calculated identity, we can now expand our math to calculate percentage similarity. This formula conceptually builds on the previous one by giving partial credit for mutations that preserve the chemical nature of the molecule. S represents the percentage similarity. We still use n sub ID for exact matches, but we add n sub sim, which represents the number of similar substitutions, where an amino acid mutated, but the new one has the same size or charge. The denominator L remains the total alignment length. Because we are adding these similar substitutions to the numerator, similarity will always be equal to or greater than identity. Let us apply this similarity formula to the exact same peptides from our previous example. We already know the total length is 5, and we have 3 exact matches. Now, biochemistry tells us that valine and isoleucine are chemically similar, and threonine and serine are also similar. This gives us 2 similar substitutions. We add our 3 exact matches to our 2 similar substitutions, giving us a total of 5 positive scoring positions for the numerator. Dividing 5 by 5 gives exactly 1. Multiplying by 100 means the sequences are 100% similar. Physically, this means every single mutation preserved the chemical nature of the amino acid, strongly implying they perform the exact same biological function. So, we have seen how to calculate these percentages. This brings us to an important question. How high must the percentage be to actually infer homology? In computational biology, for sequences longer than 100 amino acids, we use established thresholds. If the identity is greater than 30%, they are generally considered homologous. If it is below 20%, they fall into the midnight zone of non-homology. However, the ambiguous range, strictly between 20 and 30%, is known as the twilight zone, where sequence identity alone is not enough to make a definitive conclusion. Before we move on, let us see this threshold concept with a concrete example. A researcher aligns 2 unknown protein sequences that are exactly 150 amino acids long. The software finds 35 exact matches. We are asked if these sequences are homologous. First, we substitute our exact matches and total length into the identity formula. Dividing 35 by 150 yields approximately 0.2333. Multiplying by 100 gives a final percent identity of 23.33%. Physically, because this value lands squarely in the twilight zone, the relationship is too distant to be certain. The researcher must look at 3-dimensional structures to prove shared ancestry. Now that we have calculated and evaluated these metrics, let us visualize how they connect to evolution. On the left of this diagram, you see 2 aligned sequences. The solid vertical lines represent sequence identity, the exact letter matches we counted earlier. The dashed lines represent sequence similarity, the conservative substitutions. These observable alignments are what allow us to look to the right side of the diagram. The branching tree shows an ancestral node splitting into lineage 1 and lineage 2. That ancestral node represents homology. We cannot see the ancestor, but the identity and similarity on the left give us the evidence to infer the homology on the right. To wrap up our lecture, let us reinforce the most important ideas we have covered. Always remember the fundamental difference between observation and inference. Sequence identity and similarity are mathematical measurements of resemblance, always expressed as percentages. Homology, on the other hand, is the biological conclusion we draw from those measurements, stating that the sequences evolved from a shared ancestor. You can measure similarity, but you can only infer homology. By mastering these calculations and thresholds, you now have the foundational tools to decode evolutionary history. Great job following along.